Hello. Hello everyone. Today we're gonna be discussing the topic of solving addiction with nanotechnology. My name is Harrison Lin from Portola High School.
I'm Ishan Sharma from Miraloma High School.
And I am Joshua Stevens from Walnut High School.
So what technologies and nanotechnologies can scientists use to alleviate patients from drug addiction? Drug addiction has been a common issue for many years now, and the abuse of drug use has caused many detrimental, long-lasting, and permanent effects to an individual's health. Scientists have figured out ways to slow the process of people getting addicted by inserting siRNA or small interference RNA into the body of the test subject, later nullifying the DAPPR32 protein, which will be explained later on in more detail. Instead, with the use of two-proton excitation and confocal microscopy, we will investigate how inserting ibogaine will affect the DARPP32 levels. So, what actually is an addiction? Addiction is a complex condition, a brain disease that is manifested by compulsive substance use, despite the harmful consequences. For this presentation, many drugs have different side effects, but all cause the brain to secrete the well-known hormone which is also known as dopamine. And around 21 million people in the United States alone were addicted to substances such as nicotine, cannabis, etc. And there is a specific reason. In the brain perceives two systems as liking and wanting, and they view this as two separate psychological experiences. Liking refers to spontaneous delight, while wanting refers to a desire for more. In this case, certain drugs acts as the warning mechanism, which causes people to crave for even more drugs and decreases the tolerance level.
So let's talk about some history regarding addiction as a topic. So addiction was first surfaced as a disease since in the early 19th century, and the concept of addiction at first had many people doubting the legitimacy of it because there wasn't enough research following up on the claims made for this disease. However, there were some examples like the Opium War between China and Britain, which created a sense of realization that there's actually some danger regarding addiction. And finally, in 1997, the ASA, which is the AMA, which is the American Medical Association, and other medical organizations around the world, finally officially made this a disease. So our process is complicated but simple in a way. So we have this three-step plan: examination, testing, and conclusion. So examination, I'm not going to go into great detail as we're going to be explaining in further detail in the further slides. But here we're just going to talk about examination. Is basically we're going to be creating some samples and testing out some, and then we're going to move on to testing out these samples, and then finally we will be finalizing these results.
So how are current scientists approaching this problem? So we have a case study that shows how scientists tried to use gene silencing against DARRP32. What they did is they basically took siRNA, which is small interfering RNA, electrostatically complex to golden nanorods, which would be transferred to the targeted areas affected by DARRP32. So in this figure, the host staining is used to show the siRNA. So in figures A and C, C displays normal siRNA, while A shows the golden nanorod complex actually being successfully combined together to the siRNA. And figure B and D just shows regular DAN cells. So in this figure, we have a positive control, the new complex that they were experimenting with, and another positive control. Obviously, the negative control siRNA was expected to have no effect on reducing the amount of DARP32 present in a human. So obviously, there's no results there. As for the siRNA nanocomplex with GNR, it showed a 85% effective reduction rate in 24 hours, and 87% reduction rate in 96 hours, and I can't read that from here. What does it say?
81%.
81% reduction rate in 96 hours or
One week.
1 week.
Yeah.
In 1 week. Meanwhile, our positive control is actually another product that's already been released that has similar effects to the new nanocomplex that these scientists are experimenting with. However, as you can notice, it is much less effective, and it takes more time to reduce the amount of DARP32 present in a human. And then these results show how much DARP is present in a human after 120 hours of being in a human body. So the negative control shows that there's no change whatsoever in the amount of DARP32 after 120 hours. Meanwhile, the new nanocomplex that the scientists were working on, it shows an effective 67% reduction rate of DARP32 in the human body. However, and then its competitor product, which is the positive control, only shows a 29.7% reduction rate.
So let's move on to the project proposal. So as we all know, previously stated that addiction is a relatively new disease, relatively to other diseases out there. So the idea of the relationship of nanoscience and addiction is really new. So our group itself has developed our own hypothesis and solution that can, in theory, might be able to help treat addiction as a disease. So we will be using platelet cells in the blood. We'll be using platelet cells in the blood plasma. So blood plasma, as we all know, carries proteins and is around our whole body. So a company called Platelet Biogenesis, so then they have this idea of using platelet cells inside these blood plasma and use transfusion as a way to spread this cure. So we're going to be inputting an alkaline called ibogaine. So ibogaine is something that rehabilitation patients use when from cocaine or heroin. So we will be using two photon microscopy, which is a relatively new microscopy compared to other techniques out there. And also the confocal microscopy to observe the individual platelets cells through the process of eliminating DARPP32. So jumping back to the platelet biogenesis, their idea was to, from human-induced pluripotent stem cells are poised to become among the first stem cell-derived tissues ready to clinical use, representing a major first step towards a sustainable donor-free blood system. So the hypothesis. Our hypothesis is we will genetically engineer platelet cells inside blood plasma with ibogaine, which will specifically target the DARPP32 and it will act as antibodies and eventually eliminate all secreted dopamine. And the microscopy imaging techniques. So we will be using two different microscopy techniques. The first one will be the confocal microscopy, as I mentioned before, and the two photons microscopy in the research. So the confocal microscopy will provide us 3D images regarding how the ibogaine is countering against the DARPP32. And the two photon microscopy is obviously it can look into deeper tissues and the DRPP32, so we can look at how the relationships has changed and if our hypothesis is correct. So some social context about this. So by doing this, we will develop methods to combat addiction and learn more about neurological disorders because in the science field we haven't fully understood the whole science behind the brain. And there will also be potential new cures for other diseases as well. So some future directions. The platelets can potentially target other kinds of diseases as well, not just addiction, because as we all know, the platelet cells can carry many different forms of treatment, not just ibogaine. And yeah.
Okay, so overall, the usage of platelets in blood plasma as a carrier for certain elements or proteins could be a solution to our problem of drug addiction. By using ibogaine to target DARPP32 in the brain, we can lower the levels of dopamine within the brain itself and decrease the occurrence of the patient of becoming a drug addict. We would use certain microscopic techniques, as mentioned earlier, such as two-photon excitation microscopy to image the action occurring within the brain, allowing us to conduct our research. However, since this is a viable solution for solving just addiction, we can theorize that we can use the same theory, not just to solve addiction, but for more elements in the future. We can use the same procedure to target different parts of the body and certain diseases which we could not yet cure and will be investigated in the future. And the Brain Initiative seeks to understand the basis of our own brain, and by using technology and nanotechnology to seek these sites of the brain. The Brain Initiative has been successful using nanotechnology and technologies such as certain microscopes in investigating common brain diseases, and now we have understood more of the brain overall.
So the contribution, just like Ishan said, this will be able to help cure other diseases as well, potentially. And since we have such a limited information regarding nanotechnology and the idea of addiction, we will be able to do more research and eventually have more info regarding this. And some challenges. So we will be facing some challenges. As we all know, addiction is in living samples, so like humans, and we can also, but at first we're gonna be testing out these examples in like mice or other animals out there, and because we cannot input this technique onto a human without actually testing onto it first. And also delivering light to the brain. As we all know, microscopy, we use several different techniques, and we usually input light into the brain so we can look carefully. And these are the references. Thank you.
Okay, so I've got a question. Are you guys worried about the effects of drastically decreasing the amount of dopamine in one system, like depressive effects?
So you're saying if it's gonna be overkilling the dopamine?
Yeah.
So so based on our research, the blood plasma cells, we can control how much we input into the platelets. So then we can analyze how much the dopamine is over the normal amount, so we can control the amount how much we put in, so that we would notify it, basically.
Go over here and then over there.
So along the same lines, you explain how you will use platelets as a carrier. How do you envision the delivery? How would you deliver your drug in the place where you want it to be delivered, rather than having it everywhere in the body?
So as we just said, we're going to use transfusion as a way to get into the body. So right now we're still discovering, in our research, we're still discovering different ways for us to input this into the body. So some ways is that there's so we can genetically engineering them so that they'll be staying in the neural side of our human body instead of going into like other areas of our body by genetically engineering them and changing their structures.
Gentlemen, very bold initiative. Obviously, something that affects many, many people. To answer some of the question, I guess if you were really wanting to look at something that affects the dopamine areas, perhaps instead of doing transfusion, something like something that's directly injected into the CNS space through like a lumbar puncture or something of that nature. But my two questions relate to the confocal microscopy. When you use that and you inject the ibogaine or the DARP32, does it look different under the confocal? Those that are affected with the DARP32 or the ibogaine, do the platelets look different under confocal microscopy?
So on the case study that we showed the case study, the scientists that did the case study, they used siRNAs to notify the DARPP32, and we are using the blood plasma cells. So if we look back on to the charts that we showed, we can see that after the siRNAs were in contact with the DARPP32, their structures were broken down from the siRNAs. So we can obviously we have our future directions. We have to be testing this, so we can hypothesize and theorize that it can have the same effect because ibogaine is basically slowing down and eventually not curing, but slowly treat the patients off the temptation of drugs. So we can theorize that the structures will look the same as the siRNA. Yeah.
I guess it relates to the question was, what are the unintended consequences of suddenly dropping, inhibiting that? Are there, other than dopamine, are there other effects that you find anything when you were researching or reading about that? Are any other effects of this particular drug, ibogaine?
I don't, not as we know of in.
I think, in my opinion, basically what dopamine does, it basically triggers like your level of happiness, and basically dropping the levels of dopamine too far will probably lead you into depression. And maybe if you're like in a depressive state, it might cause the person who was already addicted to drugs to take even more drugs. So we have to be very specific with our measurements in order for this to actually work.
So just like Sean said, that's why we're gonna genetically engineer them into the right amount needed.
Thanks. Very, very interesting.